This morning, challenges, standardizing, and post-production state, complexity, animals, and the third option. We're going to have the next phase of fun. So both expansion of fun and the real next phase of fun. So this is really the character question we have. Because as I said this morning, that we are now, I would say, in the first phase, trying to obtain the biological reference of the main species to which the genome, let's say, sufficient. But at the same time, there are many samples, and data sets starting to accumulate, which may be applied to different developmental stages in the logical order. And as we see also different risk populations and so on. So in many ways, we are already going over to the second stage. We have to do some work there and take some interest. It's the idea. So in the past weeks, we proposed a series of questions to release as a committee. And we got some suggestions. So there was the first debate, many people who actually come here. So and here is a kind of first page of potential questions that we propose today that we can discuss. The order for this arbitrary, mine, and try to put it following my mental mind scheme. So, proposed topics for what is open issues of expanding set of fung animal tissues and assay collections is first, and is really the first question, I think, as also Johan and Hans, I think, agree. What are the core samples for the core standardized assays? That means all the assays which should be done on few samples and X tissues, but really on the same. Oh, thank you. Thank you. So, we should talk about sharing samples that we have to agree on for a limited set. So, few samples of which to make all the core assays, while there are also people who want to make few assays on a large number of samples. Okay, so it's the horizontal and the vertical axis. And these reflect different priorities, different needs, different objectives of each individual group. And most probably this kind of decision to define core samples is something to do on species by species basis. Then I will go through the questions and then so you get the general idea and then we go back and we decide where to start. There will be one of you starting. How to prioritize the new tissues and, for example, which state. So there are also technical questions which are posed, like frozen fresh tissues, dissociated cells from tissues for the fung collections which are being added now. Also very important, how to define the reference samples to be used across labs, which can be a hub tissue, which is used by everybody, could be a cell line, could be many different things, but it should be a reference sample, which is also a very, possibly the best way to also be able to handle the heterogeneity, technical and biological heterogeneity, so that we need to be able to design experiments to capture, to remove confounding factors. So with references, we can achieve much better this goal. The set of questions are which standard protocols for assays are being or will be developed first by any new contributors of fung collections. How to provide access to existing sample collections, because we discussed quite a lot already, that there is the principle of sharing, where it's an open community and so on, but samples are something concrete. And if we are repositories which can share samples, it's important to know. Because these, why going to sample again if you want a reference which exists already and you can obtain maybe a piece of tissue or a cell line or whatever by a colleague. How to collaborate further and better with other committees, what can be improved. It's very important. Well, this morning, for example, I mentioned biosamples, which is a very important tool, which is developed by the metadata committee and will be available pretty soon. With the bioinformatics and data analysis committee, they are very important to us to help defining criteria and data sets that we need to use to assess, for example, if certain pipeline is appropriate for a specific assay. And so on. For these open issues, there are many other ones possible, maybe other questions which are even more important than this one, in your opinions. And we are here to listen to this, for hearing your ideas. Then I tried also to put a session on broader issues, which is, maybe should come first, I don't know, really. Moving forward to the next fun phase, which is, of course, which topics areas should be prioritized for the next fun phase, which is a huge question for which we had several inputs. And in fact, I was also wondering a while ago, should we really attempt to prioritize the next fun phase now? Or should we really consider it like something which is extremely wide and will develop maybe in different specific lines of research? There are questions on the translational impacts of FAG work on animal production, and of course, how to stimulate integration and development for future collaborative projects. And finally, we will have to prepare a synthesis for the way forward session. So this is what I have for this start of this session for the moment. I don't know if my colleagues have something to add right now. Otherwise, we can try to start and see where we end in the time we have available and reorganize in a dynamic way following the inputs. For example, I ask a question, how many here who are not yet contributing samples data sets are planning to contribute samples, sets, and data sets? Quite a lot of people. Excuse me? Yeah. That's a good question. Separating data and samples. It's not so obvious, in fact, because we heard from John Stam that, yeah, in principle, you have frozen aliquots, let's say slab frozen, which is the one which is really the most universal use you can do. And then you can run all the possible assays. But it's not so obvious. If you want to make ChIP-seq assays on frozen tissues, in fact, every frozen tissues more or less needs a specific optimization. That's why at the end, it's much easier, or quite easier, to work with soft tissues, which can be dissociated before freezing, for example. So you can cross-link the cells and then freeze, or can be fragmented after freezing in a more easy way. Other tissues like muscle, for example, are much more difficult. So if you start right away with frozen tissues, unless you are really an expert, which is possible, of course, I'm not, but I guess they're very much experts everywhere. You could be in troubles. So maybe you can think of, okay, so maybe I should use a reference sample which is from blah, blah, blah. But in our hands, at least in my project, we found it was quite complicated to say, okay, we freeze tissues and we start from there. Well, first of all, you can have frozen aliquots. You can have frozen cross-linked nuclei to share also. And then at the parallel time, you have, and then also you have primary cells. Well, this is a bit more difficult because it's a limiting amounts in general, okay? But that you can also share frozen tissues because in the meantime, somebody of a fan community has optimized the protocol for example, making the cross-linking and so on the frozen tissues, which is something which, for example, in the UC Davis, we are doing, at least for some tissues, it's going on. So if we talk of reference samples, somehow you find a way to share. We talk of reference samples, for example. It has been discussed quite a lot of time that liver could be the hub tissue. Okay? This could be a good idea because liver is soft. It's relatively homogeneous. So you can freeze, and usually it's abundant. So you don't have a limitation of size of a sample. You can make different aliquots. And many people are working on liver. Everybody so far. So everybody who is making has already shared samples. Almost everybody has put liver in it. Yeah.
It seems we are jumping a little bit around now, but since you started to talk about reference samples, we heard this morning that some of our keynote speakers told us it's not really very important to have reference samples in this functional annotation. It was very important when we did the genome assembly, but I don't really see the use of a reference sample here.
Yeah, thank you, you did.
Please don't misunderstand me. You need reference samples. But my point was that you should not limit yourself just to those. But it would be impossible to interpret the quality of the data that individual labs generate and also provide that anchorage without the reference samples. So across the consortium, you must agree on a minimal number of tissues that everyone works on and then allow people to have freedom to add to that on top. I would say the same for the assays. You need to agree to a minimal number of assays which are core, and I suggest easy. And then if you have more complicated assays, you can afford for that to be on smaller subsets of tissues.
Yeah, for example, what I understood, yes.
I just wanted to add some sort of principles just to bring everybody back. I mean, a lot of what we were thinking about is based on what you and Bernie had suggested. You know, if one axis is the number of assays and the other axis is number of samples, you know, we do, this is actually to get people more involved. You can do your favorite samples. You have to agree to do the core number of assays, but then there's also going to be a core number of, there's going to be some samples that would be measured very deeply. We sort of have to agree upon, although there's no rules saying this, but we should agree upon that. The other way to look also on sharing samples, I mean, this might be unique to chickens, but we've selected highly inbred lines. So in the case that we need to go back and we didn't isolate all those things, that we could have the opportunity. So that's, again, another way to think about it. So also within, since we have another dimension that Ewan didn't have, is we have more species, we probably should agree upon, at least if possible, core number of samples. You know, Graham's really been pushing me on, and I agree on this, that we should have the ability to maybe do comparative work. Again, how well the networks are robust, I think that's a reasonably good idea, but we should try to agree upon, at least in principle, you know, what are the, if you're going to do a project and you're going to contribute, what are the minimum number of tissues that you should take, and what are the minimum number of samples that you should do. So maybe that's the way to think about it.
So along those lines, as far as standardizing core samples, I've kind of always had this question, this concern about actually standardizing the location of each sample. So for example, if we're comparing, a tissue in chicken versus a tissue in horse or cow, it's going to be very different how much tissue, right, and which component. So how specific are we getting? Because to me, cerebellum is not that specific. So.
I think it's most important first that you select the tissues first and then go into detail. So make the core set of tissues and later on make a clear selection of where you're going to sample, for example, your kidney, and do it over species. Because indeed it's important that you will have more or less the same sample if you want to compare. And that's, of course, Faang is also having this comparative component in it, which is important if you want to compare something that you're comparing more or less, because it's always quite difficult to be precise, but you want to have, you can make it quite precise by giving instructions what's the best part to take the sample.
Well, honestly, this is really complicated.
No. I said, don't say it's easy.
For example, in our project in France, there are kind of groups of experts for each, let's say, nervous system and so on. And even only for RNA-seq, depending on the aliquots you take, for example, from the cerebellum, maybe you get completely different things. So these people wanted to take all the aliquots, mix, and homogenize. And then I said, no, because it's not what you want to do. And if you make this process, then I cannot make the chromatin assays. So there are all these discussions going on. And honestly, I think that this complexity cannot be overcome unless we make a real critical mass of data. And all this finally is in biosamples. Because then you can really sort the data and the associated data set and get ideas of what heterogeneity you really have. I don't know how to sort this out differently. Even the fact that maybe you take the liver from a chicken and you make it exactly in the same way, and I do the same in the same line of chicken in the same way, but maybe for some reason, my chicken has found something to eat before, and you take the liver, it will be completely different for a full set of genes, okay? So there are also the environmental condition, how the animal is fed or fasted, etc. So we can say, yes, of course, usually at slaughter, animals are fasted, so they didn't eat for many hours. But this is probably different between conditions. It is different between countries. It is different between commercial and experimental, and etc. So I think this is the critical mass of data which will help. And for the data sets which are associated to these samples, there I really think we need the references. We need absolutely reference data sets.
Might be worthwhile. I'm pretty sure you did it, and Wei-Jin, they also took pictures. I mean, nowadays you just try to do the best you can, but we're lucky you take pictures easy and you do as much as you can. Not so simple, but just try to do your best.
Or, for example, the speed for the sampling. It's another issue. Again, if you're very organized, you go fast. If you're less well organized, it may take hours, and then God knows what you got.
So as we try to decide this for cross species, do we have 5 core tissues? Do we have 10? I think,
Well, in our case in France, we are doing too few tissues, but we can have all the tissues stored. And for the issue of frozen, fresh, and blah, blah, we have at least 3 tissues, soft tissues, which are also dissociated in cells, so they are preserved. And we can make also chromatin assays after storing, okay? Yes. Ah, okay.
I think at least you have to do the snapshot frozen, but if you have the possibility to do the RNA later sampling of your tissues also, I think it would be beneficial to have that, because it's much more stable later on.
Yeah, that depends a lot on the facilities. The protocol that is online for the committee is a procedure for RNA extractions, which works equally well on RNA later or SNP frozen. So I think it's a big group.
I would say the obvious, but kind of think out of the box a little bit here. There are so many ifs at the moment, okay? There's so many uncertainties before we actually can be really concrete about deciding on what you want to do, right? This is what worries me. It's like, do we freeze, don't we freeze? Now, which samples do we actually take? When do we take them? There's a whole series now. Do you take RNA later? What can we get a sample that is suitable for I don't know, 5 assays? Okay. Do I need to prepare that sample in different ways to be able to do those different assays, or can I get them all in one way? There's so many ifs. It's like you need like a 6-month period and then come back and report, say, right, we've actually tried a whole bunch of these different things, and this is what we've decided. These are the recommended ways to do it. The other thought is why are we talking about doing this? So many things, because it's all about money, isn't it? Yeah? It's about funds. We can't afford to do all these things. If we have plenty of money, like in some of the NIH projects, we'd be fine. We could probably, each of us could probably do a whole bunch of assays on a given tissue, because at the end of the day, that talk last night about the correlation of different types, RNA-seq and everything else, that works with large numbers of samples and all the variability in it. And in a sense, who cares where they all come from? The more variety, actually, the better. But unfortunately, that costs a lot of money. But the variety is great because it lets you correlate this DNA's hypersensitive site, for example, and the expression of a gene down here. So if you limit that, then you can't do that kind of study. So I mean, there's so many ifs.
But you want to have a core set of tissues from a certain number of individuals, where you, as much as possible, all the assays which are possible. And you want to do, if you have an animal, you want to do as many of the core sets, but for the basics to know, okay, how different tissues will perform in different assays. You want to have a reference of this individual for as many as different tissues possible to do all the assays. And then later on, if you want to do a specific analysis for 1,000 individuals for one assay, you have a reference where you can go back to say, okay, this is how you reference individuals or reference individuals behave in this circumstances. And this will come as an adult stage, this is an embryo stage, at least I think that's the basic thing where we have to go to. But now it's still open, and we don't know. And you can't do 1,000 individuals for all different tissues for all different assays. That's why you need this core set. Now the core set, of course, is the most important, and it's preferable of those animals. You want to do more tissue to know what's going on.
Yeah, I mean, about collections, one thing also to consider is that indeed for animals, to think that you can store the tissues differently than frozen, it's probably utopistic, right? So you can pre-treat samples before freezing for some cases when you are trying to set up standards, methods, and so on. Otherwise, it's not feasible. I mean, you make collections and you have to freeze, right? Yes, right. So this is a condition. Then we discussed about, of course, the methods. So the people who are carrying out these studies now, they have to set up the method. For example, ATAC-seq or HiC or even ChIP-seq, actually. If you have a cell line in parallel to test it, it's much easier. That's what we do, personally, you too, right? Everybody's trying to get accustomed to the method by using the simplest case. For ATAC-seq, it's a lymphoblastoid cell line, which was published for datasets and so on. Because we are learning, at some point, we should think that the standards are done. But then, how many people are participating to this? Okay, because this is a big project in itself. So even to make the biological reference on few tissues and many assays, it's a lot of work. That's the issue, yeah.
I can't help thinking you're making this too complicated, more complicated than it needs to be. And I guess one simple question I have for you is, are you suggesting that every lab individually prepares material and then the data is pooled, or are you going to resource a small number of labs to actually generate the data for you? In which case, you don't have to skill everyone up across every technique.
Into this, I mean, I think that to make the reference, biological reference, it's not that everybody should contribute to it. If I agree also your point, it would be impossible, it would be awfully expensive, really a waste. Yeah, because at the end, probably, I mean, we really need to reach the stage where most important tissues, well, most important, to define what is the most important tissue is impossible, okay, let's say. A list of tissues are really annotated. And then many things happen, okay? What happens on the other side now in Faang is that many people are putting collections, which are different breeds, different conditions, and so on, which is a richness also. But of course, when you put many samples, you will do less assays. And what is the goal? I mean, it's to make use of a functional annotation obtained before so that you can choose then what you want to do on this assay, of these samples, or is to remake the wheel again. That would be, you see? I mean, so I think the best situation with my proposition is that there are the pilot projects going on now. They work to obtain the biological reference. They have to standardize the methods, okay? And possibly they share some samples. So like reference samples, hub tissue, and these kind of things. But I don't know if this is something which is shared by this community or not. It's like to really make a distinction between the horizontal axis, you want the reference which is Faang, and the vertical axis which can be many things.
Yep, so we've just been talking there about the reference individuals and the reference data sets. I'm not clear who actually is doing so for each of the species. Has the decision being made as to which animals or which labs is doing the reference individuals? So that's really one of the first things to decide, isn't it? Yeah, because we're at the stage now where we're doing a new reference sheep genome assembly on a new individual, Rambulae, to be the reference, which we hope will be the reference individual or one of the reference individuals for the Faang consortium, and we need to collect samples in the next few months, and we're not sure how we're going to do that, and so we need to make a decision fairly soon. So for sheep, fairly soon about that. Yeah, and hopefully.
This, your idea is the reference for the sheep, okay? And you are confident that you will have the resources to do this and to deliver this for the core assays.
So we're confident that we can deliver the genome assembly and we can deliver the samples. We don't have all the funding in place. We only have a small amount of funding in place from various sources to start generating the core assays on a subset of those samples. So we definitely need more funding for that.
how about you put funding aside for now? And I know that's an easy thing to say, but what you're doing at the moment is planning the goals of a consortium activity. And if you take those goals to your funding agencies, then you've got a wish list of what you want to achieve. I think if you start with the funding that you have available today, you're not going to achieve the consortium goals. So I would put I know it's important, but put those outside of today's discussions and just agree on your ideal setup.
I think in your case for your sheep is that if that animal is decided to do the core set and somebody else want, for example, want to do the lymph node, if you also collected that, then people can go to you and say, can I just take your lymph nodes sample and do the assays? For example, whatever assay one of the assays which have been proposed, I want to do the assay on my animals, but I will also include this animal or this lymph node, for example, sample, and then you will gain as a consortium, but also for yourself more, because in many cases there is no funding for doing everything, but at the end there will everything of this reference or reference animals, and there can be a selection, they are done by the community, and I think that will be the effort of a joint thing is also possible.
I might have this wrong, but I think actually the core assays are fairly well defined already. I think the core assays are well defined, and actually I don't want to get into language of reference animals. I think that's a misnomer. There's many animals. There is not like a sequence. So I don't think there's a reference sample. What I think we have to agree upon is what are core tissues, like Christine's saying. So should we get some, immune cells, muscle cells, probably things that we're going to have to be relevant for agriculture because those are driving things. But I think we can probably agree upon a certain number of samples that are relatively easy for most groups to get as a core, and then of course then you're free to get as much as you want. But I think at least that's how I would guide things. I think the core assays are defined, at least in my mind, for the most ones, and maybe we have to just agree upon the core tissues. Again, driving the minimum water work and also allow comparative stuff to do further. So that's how I look at it.
Although core assays may be defined, I think one of the things we need to think about, and I think that was what Christine was saying, is that the idea that Brian collects a whole range of tissues and he sends a tissue randomly to Richard, for example, to do lymph node, to do a particular assay, is not a good model. I think the good model is that Lab A is an expert in DNA sensitivity, Lab B is an expert in methylation, and the lymph node from Brian goes to that lab to do that particular assay. Not that we have all the labs set up to do all the different assays and we have variability that is completely uncontrollable.
Because they're robust. And so that's what we're hoping, that you don't have to ship it out. There are a few assays like the hypersensitivity, those aren't so robust. So those are being sent to key things. But for the most part, they're looking at assays that are very robust that anyone in the room can do, and that reduces the variability as much as we can hope. So that at least, I think I'm capturing it right.
Yes. Well, I have two comments. About the core assays, I agree, with some exceptions. For example, you can think that DNA-seq now could be replaced by ATAC-seq, which is much easier and so on. But it's still a new method, so there is optimization ongoing. But let's say that this could be the replacement, okay? A reasonable replacement for the hypersensitivity assay, okay, for core assays. For core tissues, I'm not sure that we have really a straight way or a method to say these tissues are more important than others. Do you? I don't. I mean, I know in our case, there are people who say, of course, liver, of course, adipose, of course, muscle, because we are interested in some metabolic traits. and so on. You can continue forever, and you reach 60, 70 tissues, all. You see what I mean?
I don't know what a good number is, but I'm thinking more like certainly single digits. And I mean, it's just going to come down to what's practical. And again, not every lab's perfect at getting things, so we're probably going to have to be somewhat reasonable. But I don't see the number being a double-digit number, at least in my mind, at least for core, just like the core assays. I think the core numbers of tissues has to be. It doesn't need to be comprehensive. It can be, but probably also trait-driven, at least that we can agree upon. So. Sure, muscle.
We've got to sort of forget about the funding issue and things like that. I mean, it's obviously important, but what is the goal here? Yeah, I mean, what's this going to be used for at the end of the day? And what do we need to get there to do that type of work? I mean, one of the goals I know that a lot of people are interested in doing is, I got a ton of genetic variation data, and I want to assign it to a functional element, which might be defined with a ATAC or whatever it is. Okay, that kind of stuff. That's all I care about. That's the fastest way to get to that kind of information. So we have to decide on, okay, we'll fix the 20 tissues or whatever it is. Okay, that's it. And we're going to come up with those numbers. And then we've got to go and find the money to actually do that. And that means something that Graham was bringing up last week. And we actually have to sit down for these various species that we're talking about and say, okay, well, now we decided what we want it for. We decided what tissues to go for. And we'll get together maybe as a big group, or we'll get together as, I don't know, 5 groups, 5 species, and go off and find the money. And to just go for it for those particular tissues and those set of assays. And then at the end of it, we got them, and away we go, we're quite happy. I mean, it all sounds very simple. Obviously, it's not. But on top of that, later on, I've got that information. Now I can do my other experiments. I might only have a couple assays I'm going to use, but I might do them all bunch of different animals and things like that. But it's completely separate.
Please don't underestimate the variability, batch to batch and lab to lab variability that you will generate by having standard assays. And in particular, if you are looking at comparing species, then you want to remove as much of that technical heterogeneity as possible. And I think that you could find it a very major stumbling block. In terms of consolidating yourself as a community, being able to point to key laboratories who have capacity and who are able to funnel through these standard assays will be enormously important to the funders as well, because what it demonstrates is that as a community, you've gotten your act together. So I would strongly recommend that you choose core labs to run all of the assays for a particular method in that lab, much as John Stam has run all of the DNA's hypersensitivity for ENCODE. If you choose to parcel out, for example, RNA-seq, then you need to do some hardcore validation between your sites to demonstrate that you've got reproducibility, and that's not straightforward. Huge amounts, yeah.
I agree with you. Ideally, that's the case, but I keep getting back to the funding. The workaround that we've sort of thought about, and this is, you've kind of done it. You get one cell line that's very easy, and everybody does it with whatever assay they do, and then that allows to tell how well your lab. So it sort of gives you a control to figure out how well you've done that. So that's sort of the workaround to not being. Okay. All right.
you. I've killed a lot of animals and collected a lot of tissues, and it's a lot of hard work and it's expensive. And the one thing I regret about it is not collecting more tissues than we did. And so we collect a core of about 17 tissues whenever we kill animals. And I sure wish that we'd spent the time to collect 30 or 40 or 50 tissues, because it's always easy when you get the next grant to come back and move to the next tissue if you've got that tissue. If you don't have that tissue, it's game over. So my recommendation would be to come up with a list of 40, 50, 60 tissues. And when we kill animals, we should be trying to collect all of those tissues according to a standard protocol, and then they should be ranked. We're going to start with hypothalamus first, then we'll move on to liver, and we'll spend our money for doing the assays on a tissue-by-tissue basis.
Probably the question is, let's say that you can make a collection like this for each species, but why should you duplicate it many times? So that's the issue, right? So you will...
I think there's lots of good reasons to duplicate this. So a conversation that the three of us were in this morning was looking at collecting immune tissues. You've said that we're going to collect all of these immune tissues on these animals, and that's all well and good, and they've been challenged by whatever pathogens happen to be out there in the environment in which they were in. There are ways we can actually go about looking at unmapped reads to actually figure out what those pathogens were, and that can be part of our phenotyping on this whole process. But then you have to think about, well, what about animals such as the ones I mentioned that have actually been challenged with very specific pathogens? Is it worth including these animals in the analysis? Sure, it's the same tissue, but the environment in which that animal was exposed is very different to animals that you may have exposed, and we will find different data, different information. And then it's just a question of, can I find the money to do this set of animals and add that into the project?
This is going to be extremely expensive.
Yeah, I think the answer to do we have to do it on different animals is very clear, because of course we sample tissues for different projects. We have sampled 24 pig tissue from 500 pigs, and that's a project where we look at obesity. So they are sampled for a specific reason. We also have edible cells and PBMCs and other cells. So you know, I think we all have, or many of us have, samples that are really valuable down the line, and I guess we should try to focus on deciding what's the core tissues here, and then move on, because you know we can walk around this for 1 hour more, but I think we all have very specific goals with the tissues that we have sampled individually, and of course you know that's something where you can really go to the level where you have a better possibility to collect or get from genome to phenome, because in many of these samples you also have really deep phenotyping information, and that's sort of separate from the discussion of the core samples here.
I agree, but you're not going to do all the assays on all the individuals. Then you will select.
No, I'm not saying that.
No, so you need also, on all different species, in principle, all the tissues from different stages where they have information of the different assays. And then people can just bring in later on, bring their own experiments, or they can already do it. But is that, okay, I want to do this and this and this tissue, I collected this material and I want to do this. Then you don't collect it. Yeah, that because not everybody can collect all samples, all the different tissues from all the people they have.
Define your goals. Yeah?
Yeah, but your goals can be different.
No, we as a consortium, we got a specific goals. Yeah, the use of this stuff, yeah, that's another level altogether. But the immediate goals are very simple, aren't they? I don't know how many assays everyone's thinking about, but just like I said, I'll come up with a number: 10 assays. That's it, right? We're going to have 10 assays, whatever it's going to be, right? For example, I would like those 10 assays done on my 5 species for liver, right? And that's it. So all I need to do there, so those 5 species, go out and get, I don't know, triplicate liver samples and send them off to get all those assays done. And I do the same for, I don't know, another 30 tissues or whatever. Don't worry about the money, not at this stage. That's where I'm going to go, right? And then we then need to decide who's going to do those assays, because I don't care who does them. Whoever does them properly is all I care about at this stage. Quickly, it could be a human genome lab that's doing it all for all the time. It's great. Then we get it done, and that's it. Then we sit down and work out how much it's going to cost, and you give me a quote, and then you could fund it in 2 ways. One, you could do it sensibly. Go to Genome Canada or whoever and say, right, I need some money. Or you could crowdsource it, all that crap. No, you could go out with a begging bowl and work out. Well, yeah, you go for a grant, or then we need to decide how to pay for it. And then we say, right, okay, it's going to cost $5 million or something. Right, let's go up and find the money.
Just two points. Just want to let you know, if anyone didn't take their poster down, it's going to be at the registration desk in a minute, and you're halfway through your time. Actually, a little more than that.
Yeah, yeah.
It'll grumble anyway. And one of the challenges for this community is that we don't have a big common pot that we're then sitting around divvying up and how we can spend it where we choose. You know, we're going after pots from national funds, some of which will limit you shifting it somewhere else. And if you've got a pot from national fund, you want to spend it in your lab, generally speaking. You want another body in your lab. You want to put some more sequencing through your sequencing facility or whatever. And so, yeah, for all the sort of warm words we've all signed up to in the white paper and so on, yeah, underneath the surface is that this, where's my piece of the action in this? When we talk about no, I'm just old enough to be able to tell it like it is, Jim. And we talk about all the assays, but in fact, the list we've presented ourselves with is 10 assays. So it's not a huge long list. If we had 100 tissues, well, actually getting 100 tissues from a couple of animals, it's hard work, Jerry. But to be honest, it's not really a big deal for people in this community. Having access to material is.
If we're going to kill animals, we should do it.
That's my point. Yeah. So if I was someone in a lab trying to work out how I get into this game in this group here, I'd be going spending my time making antibodies against specific transcription factors, because that's the bit beyond the core set of assays. You know, if we can just find some way through this, predict our own interest bit, we'll get the 10 assays done on 100 tissues in a fairly straightforward way. I think there is merit. I had some reservations about earlier on. I think there is merit in saying, okay, this lab runs all the high C assays. This lab runs all the RNA-seq assays. Because yes, we could have a set of common samples that pass around the labs, and you say, yeah, the results are different. Now, how the hell? What's the correction factor? I don't think there is a correction factor. You're going to say, oh, bother. The data are not comparable. That's unfortunate. Yeah, and in terms of the getting labs that are good at doing the different assays, once it hits the sequencer, it doesn't really matter where that sequencing lab is anywhere around the world. Once a sequencing library is made, they can go on the machine that's got the capacity. That's not the deal. The deal is getting to between the biological sample and having the sequencing library. That's the difficult bit. And I think the challenge for us is argue with that difficult bit has to be done where the biological sample is. But they're scattered all over the place. So yeah, there's no solutions there, but just the grumpy old man cynical perspective.
One's from Sylvie Queenie. I think he's from Australia. Fang won't be able to do everything. So yes, what are the main goals? If comparing species, pick 5 tissues and 1 lab do the assay.
But, I think we all agree that we need to have a core, we've already know our 10 assays. What are our core tissues that we need to have for each species? For Fang, we're all going to be doing our different assays based on those. But what are the core tissues we need to have as the ones we want to compare across species? Can we define 10 tissues that are really important that we should make it and, have, and do we have core labs that do that? I think there's a goodness in that.
Another one from Angela Amaro. In the goal is to generate a comprehensive annotation catalog. Duplication or triplication isn't it good because depending on the environment, you will be able to detect medium and low-level expressed transcription isoforms?
Yeah, as a matter of fact, there will be a lot of duplication for whatever is RNA-seq, much less for chromatin assays, even almost zeros for IC or more complicated technologies, which are not core assays, by the way. Right? For RNA-seq, we will have so many data sets.
Yes, sorry. So just would like to go back to the idea of reference animal, because I think there is a value on doing reference animal. Most of us were working with domestic animal breeds that are highly selected and specialized. So one of the questions that always you think, like, if your experiment has control and whatever you are studying, but you think that how this would be in an unselected animal, how would be in a wild type animal. So I see value of like generating a reference animal with which would be controlled, like an unselected or wild type. For example, in chickens, if I want to do like those type of experiment will be extremely hard because, for example, if I want to know how was this gene expression in a wild type red jungle fowl, I have to bring those eggs and hatch it and make experiments, which are extremely difficult to deal with these type of animals in captivity. So I see still like there's value to have a wild type or unselected animals to create a reference animal.
Actually, this is what we chose for chicken at INRA. We took a white Leghorn population, which is the original population where another line was derived. Okay, so it's not under selection. So we're thinking this actually. But then, in fact, you have all the possible cases. For example, in the US, they use lines, right? The 6.
Not to disagree, I mean, there's different goals. The main reason that we chose the 2 chickens, and actually it's an F1 cross, is because we took advantage of the inbred blind. Because we didn't know what we wanted later on. We wanted to be able to do developmental sampling. We wanted to limit genetic diversity. I mean, that was later on the goal is to superimpose genetic diversity, but we wanted to reduce all those variables. And so we decided to use an inbred line so that way, actually, I could ship you eggs and you could do your favorite thing. This is going to be unique in chickens, but that's the logic that we chose.
Yeah, you could also make a different reasoning. I mean, we also keep the line and we can reproduce it. So, of course, the different countries have different interests. And the point is that the pilot project started before this workshop in many ways. But anyway, it reflects the reality. I mean, if you are in, I don't know, in California or anywhere else, you will have different choice of animals because that's it.
Yeah, I think this afternoon we have a really good discussion. And so about the reference animal and actually, and also as just now we also discussed about the key assay. And hence actually, as we already well defined, case assays. So I think this is very important, just list this the website. As for the reference animal, I do suggest maybe because we have so many domestic animal species. For each species, we just choose most popular breeds, okay, just list that. And then everyone, based on the question, just collect the tissues. For example, we collect 10 tissues, 15 tissues, and based on question, and then do their best, close to that the goal, or achieve that goal. I think this is more important. And for example, we also collect the more than 15 tissues from Angus beef. And so we are doing something. So and for hosting and also for other pigs, okay, so many groups do that. I think maybe, and for committee, just collect all the projects so far are already available. Oh yeah, okay.
So, and that's something you have to start with an animal where somebody collects all the tissues, and we will do, for example, the core set of essays on, and then you build on top of that, because if somebody else will do something more duplicate, and then ask, okay, can you do the essay on your sample also? Then it will increase this number of essays done on this particular sample. That will be the reference. And of course, the reference is a stage. And then you have multiple stages later on, but that will come, that will build up in time to do this. So that, I think that's the reference will come up.
I'm showing.
Sorry, Dave's left the room, so I'll make his point again. I mean, sort of, we need to define what we're trying to do. And in my mind, what we're trying to do is we're walking into a room that's got 1,000 switches on the wall and 1,000 lights on the ceiling. And we're trying to work out which switch affects which light. And they're not switched independent, they are all switched together. Okay, so what we're trying to do is create a number of different states. And it's a statistical problem, enough different states to be able to resolve which switch connects to which light, and they're all blinking on and off, as John said yesterday. So you need to create enough data, and I don't think it matters that that data comes from one reference animal or hundreds of different animals, or one tissue or different tissues, the same tissue in different states. You just need to create all those different states in a way that you can resolve the links between the switches and the lights. I'm trying to go back to the epigenetic state or the methylation state, or what are the variables we've got in our equation. So I'm not sure that reference animals and reference tissues, I mean, it's nice, as Brian says, because if you've got the tissues that come from your sequenced animal, then it makes resolving some of the variations you've got a bit easier. But I'm not sure that it actually matters. I mean, we just need to start generating a lot of data.
Reference data sets, for sure. And honestly, I think that if you have a cell line or a hub tissue, now it's better a cell line, I would say.
If you have a cell line that you can then perturb.
You can then what?
You disrupt it. You change the way the expression pattern changes, and then you look at how you've changed methylation states and promoter states and whatever, because you're trying to make those connections.
I think a reference samples is just to be sure that you obtain a pattern which is similar to what is obtained in another lab. Nothing more than that.
I don't think you should be going across labs. I think you should be doing the analysis in one lab, or a limited number of labs, to be practical.
Well, that is something that at present nobody can afford because it's extremely expensive.
Because you're investing only in one lab to make technology work. You're not investing in everybody doing everything the same.
Another way to look at it is to say, if you have your core data set, which is generated by Haven Laboratories, labs who've already worked up the methodology and who you trust, so that for that method, everything gets funneled through, then the resources in your own lab leverage off that, and you're doing the more interesting experiments where you are doing the phenotypic perturbation. So it's not that you're not being resourced to do something, but you know that the quality of that foundation reference data is really solid because you're not relying on someone's PhD getting it to work, student getting it to work in this lab, and someone's postdoc getting it to work in this lab. You're not relying on people who cut corners because they don't understand the method or they're trying to do too much. You're funneling and resourcing the people who've already started the work up and are doing it well. So it's good economics, particularly from a funder's perspective. And in terms of reference animals, I don't understand why, if someone has the material, you wouldn't just allow them to put it in at this stage.
Huh?
No, in fact, quite the opposite. What we're trying to agree upon is if you're going to have an animal, you know, what's the minimum number of tissues you should collect, right? So that's the hard. No, I agree that it would be great to have a core lab, but you know, actually, outside of biomedical, most of the work being done a lot of times is done by a student. So the question then is, yes, you would want to, but are you going to then say, I'm going to subcontract that student out to do your, you know, maybe I'm cynical, but I don't think that's actually realistic given the current funding situation.
In an ideal world, you could get a technician in your lab and a student.
Well, I honestly, well, was I was. Well, I was showing this because this is from a grant we submit. Yeah, what? Oh yeah, sorry. This is just for example, okay? These are the tissues that we prioritized in France. Why? Because of course, I mean, they have a connection to our research and to what we're interested in. Now, this is an example to show that if you think of 10, 15 samples, tissues, sorry, if you think of 10, 15 tissues that are the priority, it's not that obvious to identify them as a community. I mean, yeah, of course you put the muscle, the liver, and then the brain, how many areas you have in the brain. Okay? You can, I mean, take a mammal, and it's not that you have brain, you have cerebellum, frontal lobe, blah, 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 blah. So this idea that you have a selected list of tissues, which is the reference, I don't know, maybe probably the US, where we have a complete set of assays on the list of tissues they chose, we will probably arrive to the same thing. At some point there will be overlapping and these differences, but altogether it will make a reference, a first set.
So I agree, there are a lot of tissues to choose from, but I think we have something to go on. You know, we have the tissues that were used for the mouse ENCODE project, which I think were, you know, cerebellum, cerebrum, which I absolutely agree there's different sections. But if we standardize where we're taking them, we can still prioritize tissues. But then we also need to think about this community and what we care about. And so adipose liver, which muscle is always my pet peeve, not just muscle, which actual muscle is getting taken, and standardize that across. I think there's ways that the community can all agree to whatever number of core tissues, integrating those two pieces of information, what we care about specifically across our species and what's been done in the mouse, where they showed they didn't take, you know, 60 initially, they took what, 22 or something initially and went with those. So I think we should follow an example.
One thing I would encourage us to do, since what we're trying to do is find all the regulome here, is to take a look at the fate map that we saw last night to make sure that we have as broad a coverage of the fate map that we got out of human and mouse. Because if we want to find the regulomes for these organisms, we need to couch it in terms of what we know. Because if we only focus on one area of that lineage map that we saw, then we'll miss a whole bunch, and we won't be able to go back and get it very easily.
Silence. So maybe we should start to wrap up some conclusions, because honestly, I think that I know it makes, yeah, thank you for. Because actually, the next slide was, at the end, is always the same thing, which turns around and around. We also discussed these points already, okay? And the following one that I was thinking is, again, similar, it's connected. So at the end, we always come to the same point. So there are some conclusions that can be made from this discussion. And anyway, we have 20 minutes for that. So I will start from the simple one. Anybody who's making a collection from animals should store all the tissues in the best way. That's the first thing, because you don't know maybe if you use it or not, but you must store. So, and the number of replicates for which you do that, well, it depends. It depends on how is your system. In principle, at this point, it would be nice to have many animals, but it's unfeasible probably. In our case, we work with 4 animals. In the US, we work with 2, but that was with in mind the biological reference again, okay? So you'd say you store as many as possible animals for all possible tissues you can. So as a thing to do anyway. And then you suggest these collections, you put this information in the biosamples database. Okay, and one day maybe there will be associated data sets that will be reanalyzed together with other ones we shall collect. Second thing about standards, protocols, and so on. So I understand that it would be ideal if there is a lab which is a specialist for a given assay, and that material is distributed in this organized way to different infrastructure, let's say, which are carrying out specific assays in the best way. So it's really organized, really reproducible. My opinion is that for doing this, you need a dedicated funding. Otherwise, you don't do it. It's simple. Because what you see that you have been done right until now, it's national funding and not a big funding in neither case. They were similar, the 2 projects in size, okay? And somehow we also went for different choices because, yeah, it happens. I mean, you try also to make a plan B. So, for example, if Fang doesn't take off, then what they do with all the work which was produced, okay, this was one plan B we had, for example. So, dedicated funding, and then comes the usual eternal question. What should be the goal to obtain this funding? Because no agency at present are willing to fund a biological reference. That's my understanding. Am I correct, Anna? No? What do you think? I mean, what I retain from these discussions is that you cannot really present projects and thus to make the biological reference today. You need to produce an outcome, a test hypothesis, and so you need to go to specific biological question for which you need the reference. So, isn't it? No, I'm thinking also what the agency said today.
Yes, you were saying national funding agencies wouldn't fund reference material. Well, realise that's not strictly speaking true. So Paul Fluchek, Laura Clark, and myself, and Dave put in a bid to BBSRC about 36 hours ago for the hopefully for the data coordination center function for this project, to BBSRC's BBR call. So the BBR call, which is generally an annual one of the B's is bioinformatics, the R is resources, the other's B is biological. And in fact, that committee, which I've sat on off and on, struggles to find biological resources that, it doesn't get bids for biological resources. So at some level, it might bite your hand off if next year we put in a pitch for biological resources. Now, it's not, you're not talking huge sums of money, maybe £600,000 or £700,000, 3-4 years maybe. That's not per annum, that's total. So yeah, there are possibilities, and I think some of the NSF things were infrastructure stuff, and God, what's the EC infrastructure one? But yeah, if we have to wait until 2019 to get the first tranche of money to get the samples, then I think the whole project's screwed, frankly. But
the other thing, though, is that you're thinking about it in national, and as Alan says, there's another way of doing it. He has one. The BBSRC are going to do that, but all of those funding agencies, if they saw an international initiative that was spreading the burden of a reference, whatever you want to call it, across the community, they would fund it. The problem is, if you look nationally, they're not going to. They're not going to be interested. They want to invest in an international effort that's going to move the field forward. And that's the opportunity. And if we keep worrying about who's going to do the work, we'll never get there. And I think that's when I was always horrified in our calls, in our fan calls, that we wanted to go away from the model that ENCODE talked about of expert labs, because I don't see how you can do it without that. And we really need to think big and think international and have the whole thing. And they'll come to the table if we lead them. But they won't. If they were in here now, hopefully they're not, they'd walk away because they say you have no plan and no idea how to do it.
I fully agree. I mean, I think that the point here is that it's somehow separated, you see? So there is undeniable that we started with Faang, with the means we had, very small, if you want, national funding. And so we tried to make things, and we also got some lessons. We learned something. And now I agree. I mean, probably it's the only way, think big, but really big. So whatever means is available, infrastructure, IRC, which is a beautiful idea that was put by Jean-Charles Cavit, but let's go for it. And then in this case, you can include all the nice things. You can put the elite labs for certain technologies, all the right infrastructure. We are talking of something really big. So it will take some time. It must be put together. Also, the agencies should give feedback at the end. I mean, we should organize all this. So in the meantime,
we have to start. And
yeah.
Specifically said, don't wait. Just go, because if you have to wait for funding, then maybe if the funding is not coming through, you have nothing. So I think we just have to start, and whoever who can start, have to start.
Yes, and I think that's fine. And don't be defensive about what you've done. You've got the whole thing moving, so there's no need to ever be defensive. But we just need to identify what the simple things are that we're going to do, and we're all going to do that, and then we can go. Jerry.
So, yeah, I was going to follow on with that. So collecting, if we could agree on 50 tissues and we could spread the load around, I could collect 50 tissues on a couple of animals between now and the end of the year, and it would cost you nothing. I would fund that. It would cost me about $2,000 per animal to do that, and I'd have the 50 tissues. You just have to identify what those tissues are, Make sure you can get enough tissue, because if you start talking about pineal glands and substantia nigras in the brain and things like that, you ain't going to get enough tissue to run 10 assays on them. But if you can do that, now we spread the load among everybody in the community. Hands up if anyone else can do this. You have slaughter facilities, you have animals. Between now and the end of the year, we could have these tissues collected. It would cost nothing. Can we all co-fund generating these tissues? A bunch of us can do that.
We have some.
So we would have the tissues. The only problem is that you've now got 20 different people collecting liver samples, and we all know liver's not liver, and we're going to have variability in site of tissue sampling. I think that's a small problem relative.
Small because if you have a big critical mass at the end of samples, you will work without.
I agree.
That's the point, yeah. Yeah, I mean, I think the message is everybody can collect.
Yeah, but your liver from your cow may not be from the same site as my liver from my cow. And nobody documents that. They just say...
Yeah, although the Fang group have...
Well, it can. So we can do that.
Yeah, and some of that prep work is done.
Here's the 50 tissues. Here's where you've got to go.
Yeah. So that's right, and some of that prep work is done to help guide us. Richard's done that in the Fang community. So we would be in a lot stronger place, as Jerry says, at the end of the year, if we'd have done that. We have the basis to build the funding bit then. So that, I agree with Jerry, we should just get on with that.
Yeah. Using biosamples to show that this information is organized, is shared with the right ontologies. Only thing I would recommend, it's my point of view, 4 replicates, not 2. 2 males and 2 females, whenever possible. Otherwise, 2 males, right? Because they are the smallest possibility, because then you have an X and a Y if you are in mammals and so on. But 4, I think, is better, because honestly, for even RNA-seq and so on, if you want really good QC, I think 4 replicates is better. One gender, you mean? No, I mean, I made the example of 2 males, because then you have the X and Y chromosomes as well, instead of 2 females. But if you have 2 duplicates, usually it's 2 males or 2 females. In our case, we make 2 males and 2 females, 4. And I think it's better for many reasons. If possible, it costs the double, of course.
The only other thing to add there is that we should have the protocols before we start collecting the tissue. So how, where do you collect it? What size of sample? How do you froze it? What are the aliquots? Is it going to be snap frozen? Is it going to be put into RNA later? What do you need if we're going to do the different assays? Because there's no point in collecting all these tissues just to discover you can't do half the assays because they're frozen away in the wrong way. How does it have to be done, etc., etc. And I think
honestly, this is not. Yeah, but it will not be before the end of the year. I mean, that's impossible.
PIs from each of the species to form that group?
That's a good idea, because honestly, I mean, work a lot with things, and so exchanges and having 2 PIs.
It's not just the species, you've got the input of the assays to know the correct way that they're
but
it's not just people who know about the species.
I think it's people with a physiologist that need to be involved in that process.
Well, let's say that if you have a collections, and let's say that most of the aliquots are snap frozen, okay? And you do it in a reasonable, similar way across lab, okay? Then true, there are some assays that now we are not able to do very well on frozen tissues, but that's changing. So if you store the tissues correctly at -80, then 1 year, you can process it, because there will be a standardized protocol for that tissues, which is frozen, okay? Yeah. Well, for histone marks, it should work, Okay. DNA-seq, I don't think so. Yeah, the chromatin accessibility is the problem. Okay. But for example, but I cannot say yet if it is really the right thing. What I do is at toxic libraries, I do the first steps on the sampling side, on the fresh tissues. It's very easy, straightforward, and then you store, and then you continue later. But I cannot say now it works perfectly, I have a standard, because I didn't even see the reads. Okay. So I see only the library profiles, and it looks fine, but I don't know. So the problem is that we are too few to work on this present, to give all these answers. Oh, who is this?
I guess I was asking the same thing. If there was one protocol that we could all agree on to collect the samples and conserve the samples. So it goes back to what was discussed right now.
I should also mention that a colleague of mine, Michel Tixier-Boichard, started to write a paper about collections, and that should contain quite a number of interesting stuff, important stuff about the procedures. Because there have been discussion before starting all these things, like the size of the aliquot, and even how do you pack it in tubes? Because then even to get it out could be complicated. All these technical things. And what else we should wrap up today from this discussion, in your opinion?
Environmental conditions, like what type of environmental condition the animal would be raised. Diet is important.
You have to record on the sample you're going to collect as much as possible of all things you know on this individual.
Yeah. What my understanding is that, also from the presentations we heard, but in general you want samples, animals in the normal state, so you don't look at present for environmental stress, pathogens, or anything like that. Okay, you can get a lot of information from animals as they are. At the same time, my understanding is that we are different species, different breeds, different conditions, different environments. These collections which are going to be accumulated probably are also the results of other studies which are being undertaken. So the important thing is to record everything with specific terms and ontologies and using the biosamples database. And once all that is here in biosamples, there will be the associated data sets, and critical mass will allow meta-analysis. Excuse me? You conclude? Yeah, you conclude? Yeah.
Oh, so my question was kind of similar, but was not in relation with the amount of material, but the time, because when you're collecting from a large animal and you do it after slaughter, so for how long? So if you're trying to sample as more tissues as possible, but trying to keep the tissue as better as possible before it gets a signal for apoptosis and etc. So is there a study for the different species and for the different tissues in terms of how long? So if it passed already half an hour, would it worth to collect it or not? So I think that's very critical, because otherwise then you will have expression profiles which are not correlated to what you're studying.
Well, what I think is that because the complexity in animal sampling is quite huge, there are labs, like Ben Hayes was mentioning, that they collect all tissues from a cow in 2 minutes,
Okay.
In our case, we employed more. In some cases, there are problems, so you even can take 1 hour or more. To control completely all these conditions, it's impossible. But again, if you have the same tissue sample by many labs, and it is run, for example, for RNA-seq, and then you make a clustering analysis using, again, the biosamples data sets to organize your analysis, you may assess exactly if you have a lot of variation or not. And then at least it will be a help to overcome the technical heterogeneity that you generated. So my answer is that it would be great to be able to standardize completely all these, but I think it is impossible to be able to do that.
Yeah, but it would be nice. So if there's someone who can do that, all that in 2 minutes to learn how they do it, so that we can implement. So that was the point.
Okay, thank you. About the cell line, I don't know if I understood, honestly. You mean that we should use a cell line in what sense to overcome this?
No, I didn't spoke about cell lines, Elisabetta.
Sorry, I understood cell line.
No. I said that there's someone who can do everything in 2 minutes. I want to learn how.
Yeah. Me too, but it's what he said. Okay, thank you. But you will see also reports of all this. Thank you for participating. Ciao.
Ciao.
So we finish here. I hope we made a great job. Thank you. And now we have to, but you know what I retain? We should really collect everything and think big for for real funding and obtaining it. Otherwise, after all, we are wasting our time. Start.
Yeah.
But we started. Somehow we started. It will not be optimal, but we started.